Hey guys, the last time that we were together we were using and creating our flip book for the hierarchy of genetic material, and so we ended up creating this document right here. What we want to focus in on today are the chromosomes on this page. So we initially talked about how cells go through different processes. The normal cell process, the genetic material is just going to be loosely coiled in the nucleus, and that is called the nucleolus when it's in that form. And that's the DNA is spread out so that it can have sections taken out of it to use for everyday cell functions. Now, let's say that you ended up getting a paper cut on your finger, and you had to regenerate cells or create cells to fill that void. In order to do that, we have to take this cell and completely duplicate it. So when we're using this information right here, it's going to be really hard to copy all that information when it's just loosely bound in the nucleus. So we want to put it in a different form. We want to pack it all up nice and tight in a suitcase, get it organized, and that's when we create these chromosomes right here. So we're taking the DNA from the nucleolus and we're tightly winding up into these chromosomes. And this right here is what we call interphase. Now this is the phase that happens right before mitosis, and that is what we are going to be talking about today. So we're stopping off right here at interphase, and now we're going to focus on mitosis. Now we just said that in order for you to heal, these cells have to divide, which is what the process of mitosis is. So if you go to your OneNote notebook for the VIP 8th grade science, you will have a document in there under cell division and reproduction that's labeled mitosis, and you will see this document. And if you click anywhere on that document, you'll be able to type in on these lines, and you'll also be able to draw these diagrams as we go along. If it's not working out for you and you would like to draw this out on a sheet of notebook paper of your own, feel free to do so, or you can print it out. So we already said, what is mitosis? We already said that it is cell division. But as we'll find out later on in 8th grade science, there's two main kinds of cell division that we're going to be talking about. And this particular cell division is for somatic cells. You might ask, what is a somatic cell? And all a somatic cell are any cells except egg and sperm. Those are created through another process which we will talk about later on. So then we come to the question, what is mitosis used for? We already said that it's used for healing or regenerating. So regenerating or replacing old or damaged cells. So it's used to heal us. But also mitosis is used to grow and develop us. When we have fertilization of egg and sperm, when those two cells unite, it creates one cell called the zygote. And we take that one cell called the zygote, and it therefore splits through the process of mitosis to make all the other cells for that organism. So mitosis is used for growth and development. There's also one more purpose for mitosis which we will get into on next Wednesday. So once again, we've gone through a flip book and we said this is a normal cell process. Then we wanted to tightly package that genetic material into chromosomes, and we've stopped here at interphase. So we know that our next phase we're going to start into the process of mitosis. And mitosis has an acronym for the 4 phases, and the acronym is P, M, A, T, PMAT. So as long as you remember PMAT, you'll remember the order of these phases. Now we're going to go into each phase and we're going to discuss what happens in each phase and what it looks like. So the first phase is prophase. So we draw our cell membrane, and once again this is prophase. So we've drawn our cell membrane, and we do have a nuclear membrane in there. Remember in interphase we had tightly packaged those chromosomes and it's still inside the nucleus, What happens in prophase is that nuclear membrane starts to break apart, because if we want to take the information out of this cell and duplicate it to make 2 more cells, we have to take it out of this box of the nucleus so that we can distribute it between the 2 new cells. So we're going to draw our nuclear membrane as a dashed line to represent that it is breaking down. So one important thing that happens is the nuclear membrane breaks down. Okay. Another thing that happens, if you notice in interphase, where we took that DNA and the nucleolus and we packaged it up into those rod-shaped chromosomes, is that in prophase we don't have rod-shaped chromosomes anymore, we have those X-shaped chromosomes. And what that is we took our original strand of chromosome that we saw in interphase, and we had to duplicate that information, because if we're about to make an identical copy of this cell to replace the skin cell that is no longer there, is damaged, we want to make sure that we're replacing it with the same genetic information. So what happens is we have this original chromosome, and then we make an identical copy of that chromosome, and they're connected at the central point. And yesterday when we talked about this flip book, we said that that central point of the chromosome is called the centromere. When we have these 2 sides of the chromosome put together, these are called sister chromatids, because that chromosome and that chromosome are identical copies, so we call them sister chromatids. Now when they're together like this is all one chromosome made up of 2 identical copies of those sister chromatids. So once again, DNA is duplicated, which just means copied, and chromosomes of 2 sister chromatids form. So in prophase we have the nuclear membrane breaking down and the DNA is duplicated, and chromosomes of 2 sister chromatids are forming. So one thing that I want you to know about prophase is the prefix pro. And just like a prototype of a cell phone is the first original copy of that cell phone, prophase is the first or starting phase. The first or starting phase. Pro means first. So now that we've copied our information right here, we want to get it ready to divide. And in order to do that, we have to go through metaphase. You can underline the M for meta. So we have our cellular membrane again. And now we have to organize our chromosomes in a way that's going to allow them to break evenly, because we want 2 identical copies of the cell at the end. We don't want one cell with more genetic information than the other. We want them to be the same. So it's important that we split those chromosomes down the middle. And in order to do that, those chromosomes actually go and line up in the middle of the cell. And once again, just to remind you, the reason that we're drawing these like curly Q shapes is because chromosomes are made of tightly coiled DNA. Remember our DNA page, we showed that DNA started twisting up and winding up, like if you take a hoodie string or a shoe string and you just keep twisting it in the same direction, and it'll coil up into that chromosome. So we have our coiled up sister chromatids, and they're lined up in the center. And of course we know that those 2 identical sister chromatids are connected at the center mark of those chromosomes, which are called centromeres. And in order for these to split, they have to be connected to something. And what they are connected to are called centrioles. Centrioles. And these centrioles are essentially tow trucks or tractors. Because what they do is they send out these lines or these ropes called spindle fibers, which attach to the centromere or the center of that chromosome, and they get ready to pull them apart. So this has like a little winch system that's going to crank out, or or crank in all of that spindle fiber and break those chromosomes apart. So this is called spindle fibers, which attach at the centromere, which you already know. Okay, so once again, our chromosomes line up in the middle of cell, and our spindle fibers attach. Okay. So an easy way to remember metaphase is M for metaphase means middle. Middle, because our chromosomes are lining up in the center or middle of the cell. So our sister chromatids are still hugging onto each other by the centromere in the center of the cell, but that's all about to change when we get to our next phase of anaphase. And underline the A for anaphase, because we're about to take those sister chromatids and we're going to move them away from each other. We're going to tear them apart. So sad. So we still have our cell membrane, and we still have our centrioles, which are still using those spindle fiber tow ropes. But now we have taken this X, this chromosome, these two sister chromatids, and we have pulled them apart at the center. So we have one sister chromatid on one side and the other sister chromatid from that same chromosome on the other side. Now these things are still called chromosomes. Like this is a chromosome even though it's an X shape, and this is a chromosome even though it's just the rod shape. But we just need to know that this was in a copied sister chromatid form and this is just on its own. Okay, so once again, spindle fibers pull sister chromatids apart. Okay, now our very last phase is called telophase. So if we look right here, we have all of the genetic information right here to make one cell, and right here to make an additional cell, but we have yet to split that cell down the middle and reform our nuclear membrane. So that's where telophase comes in. And for telophase, we kind of draw like a peanut shape kind of deal. And we have our chromosomes still, they're still hanging out on either side of the cell. This set was pulled to this side, this set was pulled to the other side. And our nuclear membrane starts to reform around that genetic material because our new cells are about to be completely formed. And then we also see this little bump right here. We're about to take that cell and we're going to pinch it in half. This section right here, this little area where it's about to pinch off, is called a cleavage furrow. And that is the site of cytokinesis. So we have lots of big words, but it's all really easy because cytokinesis, all it means is cell splitting. Okay? So we have our nuclear membrane reforming. And we also have cytokinesis, which is splitting of the cell. Okay. So now if we look back, after this cell is completely split apart, we will have two identical copies of cells from our original cell. So we started out with our normal cell process with that loosely coiled DNA in there, and then we moved to interphase where we took those chromosomes and we condensed them into chromosomes. And then we started into the process of mitosis. So if you look at these cells right here, each one of these cells is going to match the original cell, because in mitosis we are making identical copies. They're going to have the same information. And so once these cells are completely split and they just start going through their normal cell processes, those chromosomes can unwind to form the nucleolus that they use for normal cell processes. And this cycle continues and continues and continues as we grow and develop and regenerate and heal ourselves, and it is a never-ending process, and that's why it's called the cell cycle. So once again, this is mitosis. This is cell division for somatic cells. There are 4 phases: prophase, metaphase, anaphase, and telophase. And we will get into this asexual reproduction section the next time we meet as a class. Have a wonderful day, and if you have any questions, please let me know.